Now that you have a basic idea of mitosis, we are going to talk about meiosis, the process of cell division for gametes or sex cells. Before we get too far into this chapter, a quick review of two important terms, somatic and gamete cells. Somatic cells are non-sex cells such as skin, liver, lung, etc. These cells are diploid, meaning they have matching pairs of chromosomes and replicate via mitosis. Gametes, however, only refer to egg and sperm cells used for sexual reproduction. These cells are haploid and require meiosis for replication. Watch the linked video for a reminder of what diploid and haploid mean. For humans, this is what it would look like in different cells. On the left is a regular somatic cell, a non-sex cell, found in cells such as skin, lungs, or bones. On the right is a sperm cell or a gamete. The somatic cell on the left is diploid, meaning it has 23 sets or matching pairs of chromosomes, one from each parent. On the right, the sperm cell is haploid, meaning it only has one copy of each chromosome, no matching pairs. In Chapter 6, we discussed mitosis, the process by which cells duplicate, producing two identical daughters. Mitosis is used by all organisms for various needs. Prokaryotic cells reproduce through binary fission, creating identical clones of the parent cell, while eukaryotic organisms like plants and animals use mitosis for growth and maintenance. With mitosis, daughter cells are 100% identical, assuming no mutations, of course. Notice the mitosis diagram. The parent cell to the far left is a diploid cell having a set of 2 chromosomes. This diploid example would be 2n equals 4, or 2 times 2 equals 4. There are 2 reds from one parent and matching blue chromosomes from the other parent. The daughter cells to the far right are identical to the parents, so they also have 2 sets of chromosomes. This works fine for asexual reproduction where there's only one parent and therefore only one cell's genetic information contributed. How then do organisms such as humans that reproduce sexually with 2 parents both contributing a full set of DNA produce offspring with only the amount of one parent? If both parents contribute 46 chromosomes or 23 sets, why do offspring not have 92 chromosomes or 46 sets? And then the next generation twice that, and so forth. The reason is thanks to meiosis, the division of gametes, or our egg and sperm cells. In sex cells, cell division happens so that the parent cell DNA is split into half of the original amount, and a process known as crossing over occurs which contributes to offspring not being identical to parents or siblings. Notice in the diagram the diploid parent cell to the far left. After the first round of meiosis, which is where the gray arrows are in the middle, 2 daughter sex cells are formed. Then a second round of meiosis occurs, splitting the number of chromosomes in half, which is all the way to the far right. So instead of 2 identical daughter cells, meiosis produces 4 haploid daughter cells with a unique combination of DNA. In this example, the parent cell is diploid with a total of 4 chromosomes, or 2 sets, but by the end of meiosis, each cell is only haploid with 2 chromosomes and no matching pairs or sets. Now, when one haploid cell is fertilized by a sperm, the resulting offspring is a combination of both parents' genetic contribution and is a diploid organism once again, like the parents were. The importance of meiosis creating haploid cells is shown in this image. These organisms are diploid with 3 sets of chromosomes, 2n equals 6. When a sperm and egg fuse at fertilization, each gamete contributes one set of the chromosomes and the diploid number is restored in the zygote. Then, as the zygote divides by mitosis, all of the somatic or the non-sex cells receive the same diploid set of chromosomes. As a reminder from Chapter 6, homologous chromosomes are a set of matching chromosomes. One comes from each parent. Each chromosome in this picture has a sister chromatid from DNA replication. In this case, the yellow set is from one parent and the orange is from the other. The circle in the center is the centromere, which holds replicated sister chromatids together until pulled apart in meiosis II. Watch the linked video for a reminder of the comparison between homologous chromosomes and sister chromatids. Meiosis has interphase as well as two main division stages, meiosis I and meiosis II. Each of these stages have the same 4 steps as mitosis: prophase, metaphase, anaphase, and telophase. Interphase is the same in meiosis as it is in mitosis. There is a G1 phase where the cell grows, the S phase where DNA replicates forming sister chromatids, and then the G2 phase preps the cell for division. It is important to note that in meiosis, even though cells divide twice, the DNA only duplicates one time. This is how 4 cells are produced with half of the DNA instead of 1/4 of the DNA. Notice in the diagram, before meiosis I, the DNA replicates producing sister chromatids of each chromosome. Then, in meiosis I, the cells divide and half of the DNA goes into each cell. During meiosis II, a cellular division occurs again where half of the DNA of each cell goes into the 4 daughter cells, 1 short chromosome and 1 long chromosome in this example. Meiosis is the first of 2 cell divisions and will follow the same path as in mitosis, but with a few important differences in the outcome. The first phase of meiosis I is prophase I. In this phase, the sister chromatids will pair with their homologous chromosomes in a process called synapsis. Also during prophase I, which is very different than during mitotic prophase, genetic material will cross over from one homologous chromosome to the other. This occurs at a site known as the chiasmata. At this site, the chromosomes are held tightly together and known as a tetrad. Watch the linked video for an animation of crossing over. After genetic material crosses over, chromosomes can be a mixture of both parents' DNA. When this happens, these chromosomes are known as recombinant. For the 2 chromosomes that did not cross over, they are non-recombinant chromosomes. Notice the capital versus lowercase letter C. The blue chromosome now has 1 capital and 1 lowercase, and so does the red chromosome. This is an example of parental genetics crossing over to the other chromosome. During metaphase I, tetrads line up along the midline, similar to metaphase during mitosis. The way these tetrads line up is completely random and therefore can produce endless variations of outcomes depending on how they are pulled apart at the end of meiosis II. To better visualize the various outcomes, here are 8 combinations from 2 sets of chromosomes that could result from how the tetrads line up. Notice in the top arrangements, blue are both on the left and red are both on the right. So, there are 2 possibilities after the second round of meiosis. All blue, labeled genetic arrangement 1, or all red, labeled genetic arrangement 2. Notice the second chromosomal arrangement where the shorter chromosomal alignment is flipped, where red is on the left and blue is on the right. This would result in 2 different combinations. 2 with long blue and short red, labeled genetic arrangement 3, and 2 long red and 2 short blue, labeled genetic arrangement 4. This shows 4 outcomes of an organism that only has a total set of 2 chromosomes. Now think about this. Humans have 23 sets of chromosomes, not 2 like this example. That means there are over 8 million variations in how chromosomes could end up in one egg or a sperm cell. Similar to mitotic anaphase, chromosomes are pulled apart to the 2 poles in anaphase 1. However, realize in this step it is both sister chromatids that are pulled to one side. The homologous chromosomes are pulled to the opposite pole. During telophase 1, the chromosomes are fully separated and at opposite poles. Because the cells only have one of the 2 original chromosomes of the homologous set, these cells are now considered haploid. They still appear like doubles because the sister chromatids are still attached to each other. Also, the arrangement of genetic material in the 2 cells is not identical due to the crossing over of genetic material. Notice the tip colors are different in the 2 cells. This process is followed by another cytokinesis and will either develop a cleavage furrow or a cell plate if the cell requires a cell wall to fully separate the 2 daughter cells. When cells enter meiosis II, they go through a phase known as interkinesis, which is similar to interphase without the duplication of DNA again. These steps are more similar to mitosis than in meiosis I. During prophase II, chromosomes condense, but no pairing of homologous chromosomes and no crossing over. Then, in metaphase II, chromosomes line up on the midplanes of the cells. During anaphase II, the sister chromatids separate and move to the opposite poles. Due to genetic crossing over, the chromosomes are not identical as they were in mitosis. During telophase II, the nuclei form at opposite poles of each cell, and cytokinesis occurs, resulting in 4 haploid cells. At this stage, each cell only has 1 copy of chromosomes and are genetically different from all other cells due to genetic material crossing over. For a comparison of mitosis and meiosis, mitosis is a single division creating 2 identical daughter cells. It is used in somatic cells as well as asexually reproducing organisms. Meiosis, on the other hand, is a process of double dividing cells to create 4 genetically unique daughter cells. It is only used in gametes of sexually reproducing organisms. To study genetics, scientists use a karyotype that aligns homologous chromosomes side by side. This image is of a human male's chromosomal composition. Notice that there are 23 sets of chromosomes, 46 total chromosomes. There are 44 autosomes and 2 sex chromosomes, X and Y, for this person. Sometimes mistakes occur in the process of meiosis which have an impact on chromosomal distribution. Notice in this karyotype at chromosome 21, there are 3 copies. This is an example of why the disorder Down syndrome happens. Chromosomes are to separate during anaphase I and anaphase II. If a separation failure happens, one of the 4 resulting daughter cells will either have an extra chromosome or will be missing a chromosome, and this failure is known as non-disjunction. Notice in the left diagram at anaphase II, which is highlighted by the blue arrow at line 2, the resulting daughter cells from line 3 are impacted when the chromosomes do not separate. One of the final cells has 3 chromosomes, 2 from the mom that did not separate, and 1 from the dad post-fertilization, while the other daughter cell did not receive a chromosome from the mom and therefore only has the 1 from the dad. When the failure occurs during meiosis II, half of the daughter cells will be normal and half will be affected. The image on the right indicates the same issue, but occurs during the first division at anaphase 1. In this example, 2 cells have 3 chromosomes, 2 from the mom and 1 from the dad, and 2 only have 1 from the dad. When the failure happens earlier in the process, as in meiosis I, all 4 daughter cells are affected. So non-disjunction is not a good thing and can cause numerous issues. A euploid is an organism that has the correct number of chromosomes for its species. For example, humans should have 2 of each of the 23 sets of chromosomes. The image on the left is an example of an organism that only has 2 sets of chromosomes and 2 copies, or diploid, for both of those chromosomes. Both humans and this small organism would be called disomic. When irregularities exist in the normal chromosomal number, that is known as aneuploidy. It can be either having 1 too many, known as trisomy, or having 1 too few, which is monosomy. Notice the image on the right. The top cell is trisomic because it has 3 chromosomes. The cell just below it would be an example of monosomic because it is lacking 1 of the chromosomes. These abnormalities are a result of non-disjunction and usually only happen with 1 chromosome in humans. 2 common results of non-disjunction include Down syndrome, also known as trisomy 21, and a less known syndrome known as Patau syndrome or trisomy 13. In both of these karyotypes, notice the 3rd copy of a chromosome. On the left image, the 21st chromosome, and on the right, the 13th chromosome. An individual whose cells have 3 or more sets of chromosomes is polyploid. This is rare among animals, but is common among plants. The polyploid trait in plants helps produce bigger, better varieties. Notice the Arabidopsis plants. The diploid is the smallest, while the tetraploid and the octoploid are much bigger variations. An extreme example of non-disjunction causing polyploidy is when all of the chromosomes for one parent do not separate. This example is 3 copies. The mom's chromosomes did not separate and were then fertilized by the dad's sperm, and is therefore referred to as triploidy. When this happens, the embryo does not survive. A few examples of trisomy and monosomy that can happen with sex chromosomes in humans are Klinefelter syndrome, Turner syndrome, and XYY karyotype. Each of these produce viable offspring, but physical and reproductive challenges may exist. Watch the linked video for an animation of non-disjunction and conditions it can cause. This figure shows common abnormalities of chromosome structure. In the first example, a portion of the chromosome mistakenly made multiple copies of the segment A, and it is known as duplication. In the second set, segments A and B are inverted. So this is inversion. In the third, a portion of the chromosome is deleted, and guess what? It's called deletion. And in the last set, a reciprocal translocation occurs when 2 non-homologous chromosomes exchange segments. The long and the short non-homologous chromosomes crossed over by accident, and this is known as translocation. Any or all of these mistakes might produce viable offspring, but they almost always exhibit physical and mental disabilities in the individual. A syndrome that can happen when a portion of chromosome 5 is deleted is cri du chat, or cry of the cat in French. Physical and mental disabilities are evident in individuals with this syndrome. Also, babies with this condition will cry in a manner that sounds like a cat, which is where the syndrome gets its name. Watch the linked video for an example of the sound of this syndrome. And now we're at the end of Chapter 7. So be sure to go back and review your end of chapter questions, and be sure you turn in your assignments by Thursday night. And have a fantastic and safe 4th of July.